over to Dr. Kazandjian.
Thank you very much, Cindy, and thank you to you and your whole HealthTree team for giving me the opportunity. And of course, thank you everyone for tuning in. I'm happy to make this interactive. So if there's any questions, you can ask me, along the way, or we can, and we can also answer questions at the end as everyone likes. I will share my screen now. Okay. So as mentioned, it's been about 4 years. I've been at the University of Miami in the Sylvester Myeloma Institute. My main role there is obviously being a myeloma doctor and really lead a lot of the myeloma clinical trials, translational work. I also have broader responsibilities in all of Sylvester in terms of really opening these trials and giving opportunities to patients in South Florida. So today I kind of wanted to talk about two specific studies that are different, and I will try to go through them slowly so one can understand the differences. But it's really trying to use immunotherapies to see if we can kind of push to the next chapter in the treatment of newly diagnosed myeloma. The two trials, the first one I'll be discussing is called the Immunoplant study, and the second one is called the ALPDUES study. So just to give a background, this is how I treat multiple myeloma, and many of us at Sylvester. As you all know, for newly diagnosed myeloma, you may receive, or patients receive, 3 or 4 drug cocktail up front. More and more quadruplet-based therapies are becoming the standard of care. For those who may not know, the quadruplets are really referring to the anti-CD38 antibody called daratumumab or isatuximab in combination with either Velcade, Revlimid, dexamethasone, known as VRD, or with Kyprolis, Revlimid, dexamethasone, ARD. And many patients, somewhere between 50 to 80% of patients receiving these treatments, do attain MRD negativity. We hear of MRD a lot, but just to recapitulate, MRD stands for minimal residual disease. It's kind of like traditionally when we do a bone marrow biopsy to see how a patient response to the treatment was, we looked under the microscope. This is basically looking even deeper than just the microscope, where we use kind of fancy techniques, either flow cytometry or next generation sequencing, to look deeper. Sometimes it's kind of like, I give the analogy, if you clean your, or if you like mop your floor or sweep your floor, you get most of the dirt, and with your own eyes you may not see anything. But then if you run maybe I don't work for Dyson or anything, but if you run a Dyson vacuum or some high-powered vacuum, you may actually pick up even more dust that you just didn't see. And so this is kind of similar to that. And the current sensitivities, or what we call level of detections, are about between 1 in 100,000 to 1 in a million. And what that means, sort of in simplistic terms, is to be able to say with scientific certainty that after looking at 1 million normal cells, for example, that one bad myeloma cell was not found. And what we do know is patients who do attain MRD negativity overall do well. There's a caveat. If you don't attain MRD negativity, it doesn't necessarily mean you're going to do bad in general, but if you're negative, then most patients, have good long-term outcomes. And what you see the slide here, this is sort of the paradigm of how I treat newly diagnosed myeloma, where patients, if MRD negativity is attained, then they could potentially forego high-dose chemotherapy and stem cell transplant into a maintenance therapy. If they're positive, then it really takes a conversation about the role of transplant and in many settings, transplants has shown that you, in a subset of patients, maybe about 10 to 15% patients may attain a deeper MRD negativity if they haven't attained it yet with the use of the stem cell transplant. That may be changing with some of the new novel treatments and some of the data that was presented at ASCO, but that's very sort of preliminary. So my really question and the question of this trials are, can we actually convert to MRD negativity with some of these newer immunotherapies and see if some of these new immunotherapies could take the place of high-dose therapy with stem cell transplant? So that brings me to the first trial, the Immunoplant study. The long title is called Immunoconsolidation for Newly Diagnosed Multiple Myeloma Using Lack of MRD Negativity After Initial Combination Therapy to Pursue Deeper Responses with Linvoseltamab and Delay Transplant. So in this study that we have open, we've enrolled about plus or minus a dozen patients, a little more actually, than a dozen patients. And it's for patients who attain the, who receive, excuse me, their triplet or quadruplet based novel combination treatment and attain a very good partial response or a complete response, which in the big scheme of things is a very good response to treatment. But maybe on bone marrow biopsy, they're MRD positive still. Those patients, as like alluded to, it would be a group of patients that may benefit from high-dose therapy and transplant. And that's where this trial comes into place. If patients, have those kind of criteria, they can enroll on this study. And on this study, they receive an immunotherapy called linvoseltamab. For those who don't know, linvoseltamab is a bispecific T-cell redirecting treatment. It is not yet FDA approved, but probably will be FDA approved in the coming months, probably by the new year. It's very similar to the two BCMA-based bispecific antibodies that are approved, meaning elranatamab and teclistamab. So once patients get treatment for 4 cycles, then another MRD assessment is performed. If patients attain MRD negativity at that point, then they're done with the study treatment and continue to go on to standard of care maintenance treatment, which in most cases is lenalidomide maintenance. In some cases, maybe the addition of daratumumab. And then if they're positive, then they receive 2 more cycles of linvoseltamab, and then would go on to the maintenance treatment. So the study rationale I have over here, in brief, is to attempt to convert patients with low level of disease by targeting the last remaining sort of resistant myeloma clones using a unique mechanism of the T-cell redirecting bispecific antibodies. As I mentioned, patients receive 4 cycles, and that's it. If they become MRD negative, at most 6 cycles of treatment. And this time frame was kind of based on some of the other preliminary data of other bispecific antibodies. Here's just a little more background on linvoseltamab. As I said, it's very similar to teclistamab or elranatamab. It's definitely has been shown to be safe and effective and pretty much in line with the other bispecific antibodies. These are key eligibility criteria. We kind of went through most of it, but as I mentioned, it's for folks who received either VRD plus or minus an anti-CD38 antibody or KRD, same. And patients have to have had at least a VGPR response to the upfront treatment, but remain MRD positive. Here's pretty standard other criteria that's needed. Nothing really jumps out. And for those who are interested in sort of the mechanics of the treatment, it's an IV treatment. It's given daily, it's given weekly, excuse me, for the first 3 cycles. and then it's given every other week for cycle 4, and then 5 and 6 if patients remain on that for cycle 5 and 6. Importantly, for cycle 1, there's a lot of you are familiar with the step-up dosing for these bispecific antibodies. The step-up dosing for this treatment is weekly, and the first 2 doses necessitate a hospital admission for 24 hours to make sure that cytokine release syndrome or CRS does not occur, or if it occurs, that they're in good hands to treat it. That was the immunoplant study. Now I'm going to move forward to the ALP-DUET study. The ALP-DUET study is really meant to target a patient population that is already receiving maintenance treatment, but either did not have a very good partial response or a complete response, or remain MRD positive while on the maintenance treatment, or for patients who did have a good response up front but had a quick relapse during the maintenance phase. And for those are the patients that would qualify. And the treatment, as you can see here, is again with linvoseltamab, but this time in combination with the lenalidomide maintenance treatment. And patients get about 2 years worth of concomitant treatment. Here's the inclusion criteria. We spoke about this already, but in short, patients, would have had to receive the standard of care novel upfront therapy. They could have received a transplant or not, and then are on the lenalidomide maintenance treatment when either cohort 1 at the time of assessment, they're MRD positive and have at least a partial response, or in cohort 2 where they were MRD negative but then had a relapse within the first year. Again, we need to have adequate organ function, which most patients do have at this point. I think the criteria is fairly open. And the treatment for the linvoseltamab itself is very similar. 2 hospitalizations, a week apart, each 24 hours. It's every week for the first 3 cycles. And then for cycles 4 to 6, it's every other week. But because this is a 2-year maintenance treatment, starting cycle 7, so about a year and a half, it's only given on day 1, so of each month. So it's only monthly. So those 2 trials were the main trials I want to sort of bring up to the HealthTree community. I think they're novel trials in the sense that we're really trying to target niche patient population with newly diagnosed myeloma to see if we can sort of impact towards the better overall outcomes. We also have other immunotherapy trials. I've spoken about this in the past actually with HealthTree, and you can probably find the webinar if you're interested, but we also have the REVIVE study, which is a study that's actually for high-risk smoldering myeloma, but again uses the immunotherapy or the bispecific immunotherapy called teclistamab, and is given in combination with daratumumab. And then we have Advanced 2.0. Advanced 1.0 was just presented actually by Dr. Langren at ASCO a few weeks ago. And Advanced 2.0 is on its way and being opened in the next coming weeks. And Advanced 2.0 is for patients who have newly diagnosed multiple myeloma and who are treatment, as we call it, treatment naive, meaning haven't received any treatment yet for it. And it'll use the bispecific T-cell engaging treatment called elranatamab, and it's going to be given in combination with lenalidomide and dexamethasone. On that note, I just want to thank you for your attention. I'm happy to answer any questions, and thanks for giving me the opportunity.
Perfect. Thank you so much, Dr. Kazandjian. And I apologize, I did not mention this to begin with, but our first question just asked about the range of MRD positive patients in the study. This is one of the open trials that's available at Sylvester. So this would be something that if you're in the area, if you want to talk Dr. Kazanjian about what it would look like to join the study.
Sure. Yeah, unfortunately, you'd have to come to one of our locations, although it is available in some of our satellite locations, but you still have to be sort of within the vicinity of South Florida. Um, and it can be difficult if you're not local, obviously. Some people have pulled it off. Um, some people just have, family or friends or have it, we're like the snowbird city here. So some folks have other housing here. But it would entail, weekly visits basically for the first 3 months at least.
Okay, thank you. And I think you answered this as well within the presentation, but just asking about administration of the bispecifics in these two studies. We had some step-up dosing, it looks like in both, and then onward to administration. Can you go over that one more time, just what the administration looks like?
But sure. So the cycle one, day one, as we call it, the first infusion is at a very small dose, 5 milligrams, and that's part of the first step up. It's given in the hospital, so we can watch for any side effects. Most patients fly through the treatment. We give a lot of prophylactic, um, medicines for both cytokine release syndrome and for infections. The infusion itself is probably only about an hour at most, but then we have to sort of, what we say, watch you for about 24 hours before you can go home. And then you come back in about a week, I think it's plus or minus a day, and do the same thing. You get a higher dose, but the infusion rate time is about the same. And then, presumably everything goes well. And so far, everything, all the patients we have had on the trial, there hasn't been any major issues. And the third dose, is given as an outpatient. And so that could be given in one of our satellite clinics or downtown, but not in the hospital.
Okay, thank you. Could you talk a little bit about linvoseltamab? Is it, you said it was pretty similar to some of the other bispecifics that are on the market?
Yeah, it's very similar to, for example, teclistamab. The side effect profile is the same, the rate of CRS, everything. I think like biologically, the only difference is that it binds that BCMA in a little bit different location, but really the end result is the same. And quite honestly, like the half a dozen or so of these T cell bispecific antibodies, they all work very similarly and have the same kind of what we call the efficacy and safety profile. So it's very
Okay,
similar. The only difference between all these is kind of like how often you receive it and whether there's a subcutaneous or an IV version.
perfect. And right now, as you said, the linvoseltamab is a IV, is that correct?
That's right.
Okay, perfect. All right, our next question just asks about will the use of this BCMA product in newly diagnosed multiple myeloma patients eliminate patients from a CAR T product with a BCMA down the line?
I don't think so. I think the verdict is still out. I mean, I think initially there was a lot of flag waving that if you get one, you can't get the other because it targets the same thing. I think, and then we see data that comes in that shows no, you can still actually have a response to CAR T, BCMA after. It's really highly dependent. I think it definitely, I wouldn't use it right after, like a BCMA CAR T right after receiving this treatment. But down the road, I think it's definitely possible. I don't think we really have enough evidence or data to suggest you kind of box yourself in choosing one over the other. I think in the older days, it was more of an issue because the clinical trials would exclude patients. But because CAR Ts are available now, many of them, well, two of them at least, then it's fine.
Right. Yeah, I know the question of sequencing has come up a lot, and I know it's a pretty hot topic. It'll be something that we'll, continue to see results on, but yeah.
Yeah, no, exactly.
All right, next question says, what do you see as the main benefit of using this bispecific as compared to currently FDA approved bispecifics? And what could cause you to choose this for your patients?
I think that's an interesting question and an important one. Um, I, there's, I can tell you, I think in my mind, it's usually sort of all the non-medical things that potentially drive decisions like that. I think medically or clinically speaking, they're all equal in my head, quite honestly. And forget about this one because it's not FDA approved. Even with the FDA approved ones, one could ask, why would you choose one over the other? And they're both FDA approved, right? So I don't think there's a major reason. I mean, I could tell you, with all my biases, um, I feel like this one may be a little more gentler, but it's completely non-scientific. I'm just disclaiming myself. It's completely very non-scientific for me to say that. It's all anecdotal. But when people ask me this, I say they're all equal. It's just the only difference is it's a different company. Choose your
Absolutely.
Nissan, Honda, or I guess whatever.
I love it. Thank you. I like the disclaimer there too. That was really important. But um, yes, some of the other questions that came up while you were talking through the different trials was with the the ALP um, Diaz trial. Is that one, have you seen a lot of interest in that one yet? Or is that one newer?
That one we just opened actually. So
Okay,
that one we have a couple of people on the wait list, and that's getting activated. But that's yeah. So that's newer.
right. Got another question that just asked, during this study, are you able to go down on the dose? Like if you saw, side effects that were um, not complementary, would you be able to go down on dose?
That's a great question. So for the ALPDUET study, to remind everyone, that's the study that is really used as part of the maintenance strategy in combination with lenalidomide. The lenalidomide dose could be changed as per standard of care, whatever like sort of one would do as part of standard of care. But when it comes to the actual linvoseltamab, the T cell antibody, doses do not get lowered. That's kind of like a hallmark of antibody-based treatments, is that there is no dose. There's only one dose that can be given. It's a pharmaceutical property of antibodies. The only thing that can be done though is you can have dose delays for like side effects and whatnot.
Perfect. Um, and with the other studies that you mentioned, the Advance 2,
Yeah,
um, could you just explain for our audience what it means to be treatment naive? Just talk through that.
it's really so we have broadly speaking, there's multiple myeloma and all its related plasma cell related conditions. So, for example, monoclonal gammopathy of undetermined significance on the one end, known as MGUS. Then you have something called smoldering multiple myeloma. Then you have multiple myeloma. And then you have other entities. There are really subsets, but things like, like plasma cell leukemia and other things. When we go down sort of the chain for multiple myeloma, we can kind of divide into two boxes. One is what's called newly diagnosed multiple myeloma. What that basically means, it was just newly diagnosed. And that's why it really implies none treatment yet. You haven't treated it yet. And that's what treatment naive means. As opposed to the other box, which the majority fit, because there's a lot more people that would fit in that box, is relapsed refractory multiple myeloma, which basically means, relapse from the initial treatment. So
Perfect.
newly diagnosed myeloma, treatment naive means pre-first treatment.
Perfect. So having no treatment for multiple myeloma. Perfect. Um, could you just describe a little bit about bispecific antibodies, how they work, why there's so much excitement around, all the new developments with different targets and things like that?
Sure. Um, so bispecific antibodies, CAR Ts also, but bispecific antibodies, they really have changed the natural history for the better for myeloma. I can tell you on the initial studies, these agents, in a very difficult tumor to treat where 6, 7 treatments have failed, 60, 70% response rates were being observed, meaning in an area where you wouldn't expect any tumors to have a response, you'd now see 7 out of 10 having a response to treatment. It is pretty remarkable. So to put things in perspective, I always use this analogy is daratumumab, right? It's a great drug that also revolutionized how we treat multiple myeloma. Its first approval was in a similar patient population where 6 or 7 treatment lines were used already, and the FDA approved daratumumab initially for with a response rate of less than 30%. My point is, daratumumab, which is a great drug, we were happy to see a 30% response rate, and the bispecifics in the same patient population showed like 60, 70%. So it really
Amazing.
how we do things, right? So
Yeah.
how this works is first and foremost, it's an immunotherapy because it uses your own immune system. It kind of like sort of um, like kind of wakes up or resets the thermostat of your own immune system. And this antibody, once it's given, it has 2 arms, and that's why it's called bispecific. It's specific for 2 things. Those 2 things, we call them like they kind of look for targets. And those targets, one of them is on the actual myeloma cell. The other target is on your own killer T cell, whose job it is normally to kill cancerous cells or malignant cells, but which in this case, for some reason, just doesn't see the myeloma cells the same way. And this antibody, basically with the 2 hands, brings them into holy matrimony, as they say, only to have a really bad divorce where the killer T cell gets activated and actually attacks and kills the myeloma cell. It's almost like a facilitator than an actual chemotherapy or cell toxin.
I love it. Yeah. I like the idea that, often when you think about cancer, one of the biggest problems is that the cancer goes undetected. Your body doesn't notice or know that it's something that should be taken, attacked or whatever. So that's just bringing those two together so that your body can take care of the cancer. That's beautiful. So you've got a question that asks for your patients that have a prior history of Bell's palsy, do you consider bispecific or CAR T? Is there any research around that?
There's no research, so this is all sort of clinical best opinion, I guess. And when you have those situations, if you ask 100 doctors, you're going to get 200 opinions. But I would say for sure, like if it were my patient, I would actually aim for the bispecifics over the CAR T because they tend to be sort of less aggressive in terms of some of these, autoimmune like they're not really autoimmune, but autoimmune like side effects.
Absolutely. In that same vein, could you just describe what CAR T looks like for those that are not yet aware of CAR T? How it's, thanks.
So I can it's kind of so the CAR T cells basically um, are man-made modified T cells of your own T cells. So in very simplistic terms, patients have their T cells collected and then genetically modified in such a way that that T cell now is like a monster and just wants to kill one particular type of cell, that whichever cell has that target, right? And that target is the myeloma, is the target found in the myeloma cell. What there's, like I alluded to, there's two approved. They're both the same target, which is why that other question was BCMA. But there are other targets like GPRC5D. We have one for, bispecific, but they're also being developed for CAR T.
Perfect. Thank you. And yeah, two questions I have left here. Number one, what are you most excited about, like in the at ASCO or EHA that you saw recently? Are there any studies up and coming that you were particularly excited about?
I think like ASCO for me, I guess because it was the most recent meeting, it was like very exciting actually. And a lot of times it's not so exciting for the sort of the blood cancers, but I thought it was very exciting. There was like one recurring theme, and that was quadruplet based therapies are really changing outcomes. We have like now 6, 7 randomized studies, including one of them is ours from the Advanced 1.0, that are showing that you know patients do so well up front now in terms of attaining MRD negativity and deep remissions, and that's the way to go. I think what's exciting for me, like our overall theme, is some of the studies that I just presented, and of course others are doing very similar studies in different contexts. But like I said, this bispecific, for example, T cell redirecting therapies, they did so well, as I said, in the relapse refractory setting. So it's only natural you move them up. But it's, and we always do that, and that's just how drug development happens. But I think one major plus in this type of therapy in this case is that since we're using patient's own immune system to begin with, it's not only that you're hitting the myeloma before it has all the new mutations and resistance, but you're also using it when your own immune system is healthier. And I think that's very exciting because I think I think given some of the responses and some of the preliminary data we're seeing is moving these things up front, we may be really getting close to functional cure in more patients. We already see that now, but it's hard to know with who and what, and the rates are low. But I think we're going to see a whole different era in multiple myeloma in the next decade.
Absolutely. You mentioned it earlier with your Revive study. I think treating high-risk smoldering myeloma. Do you think there'll ever be a day where we start looking at, maybe treating just smoldering myeloma?
This is what I think, and it may be controversial, but I don't think there's probably not any other doctors, or at least myeloma doctors on this call. Probably, I shouldn't say that unless I knew, but I actually think the field is. I'm not so sure the field is going in the right direction right now, and I think it will change. But I think we're also going this way because the lack of enough understanding of the biology underlying smoldering myeloma and multiple myeloma, so we have kind of no choice. But I think the following is the future. The future, I think, and this is the controversial part, is there is going to be no smoldering myeloma. It's going to really be more accurately defined as either stage 0, newly diagnosed multiple myeloma, or it's going to be something like, MGUS like, or MGUS maybe. And once we can do that in a robust way scientifically, then it's irrelevant to do all these studies in smoldering myeloma, because what we're all gonna do is use the best and the next greatest therapy that we have for newly diagnosed myeloma for stage 0 myeloma. It's not so different than what has happened. Probably many people don't know this on this call, but there used to be this kind of really bad term called ultra high risk smoldering myeloma. And in 2014, those patients all got sort of reclassified as having newly diagnosed myeloma based on, some people call it the slim criteria. But I think that's going to be done in a better way than just using light chains, which is not accurate all the time, and other parameters. It's going to be looking at the cancer's DNA. We're going to know.
I love that. I love the future is so bright. There's so many new options. And like you said, the testing is insane. It's amazing what we're able to do.
Yeah, yeah, for sure.
Love it. Um, Last question, unless you get some others while you answer this, would just be if somebody is interested in participating in any of those studies that you mentioned, what's the best way to get in contact with you or someone at your clinic?
That's a great question, and I always forget I should have a slide with that information. I always forget, but I mean, you can share my information. I could because sometimes a lot of times I've noticed that patients are interested but really are questions about the eligibility. So I'm happy to answer those. But if things move forward, then we would just it's for now. I guess it's best to just if you can share my email address, and then I
Absolutely.
the lines, if depending on where it goes.
Yeah, absolutely. I will share that in the follow-up email. If there's another contact or anything else you want to add to that, Dr. Kazanjian, we can communicate offline and make sure we get that all
Yeah,
squared away. I know on a previous call that we had about your immunoplant study, we had at least one patient who was watching who felt like this study was made just for her. So we just want to thank you for your time today and in sharing your enthusiasm for these new studies and sharing the information. So we hope that somebody on the call today will benefit from the information that was shared. So thank you.
sure. I will send you that person right on the chat here, okay, Cindy?
Yeah, perfect. Okay, and I will copy this real quick. Yeah, awesome.
Any other questions?
Oh, no, we didn't see any others come up. We did have several thank yous. Just want to make sure that you know how grateful everybody is for sharing your time today. So, and I just added to the chat to everyone the two things that I mentioned earlier, the um, bispecific antibody study and then also our Connect to Cures program. So if we have no further questions, we'll close out the session today. I'm just going to look one more time. Yeah, just many thanks for your time. So I think we are
Oh,
good.
and everyone for watching.
Absolutely. Thank you. All right. And I'm just going to share the results of our poll real quick. It was kind of enlightening to see how many of you have participated in a research survey before, hopefully through the excitement of Dr. Kazanjian today and introduction to some of our surveys. We hope that you'll decide to take part in a survey and help further research for future generations down the line. I'm going to share my screen again and we will wrap up. All right. So, as a note, we of course have our quick survey at the end of today's webinars. So, if you'll take just a few minutes to fill out the survey, it helps us decide what topics we need to focus on to get you the most correct information possible. So, take those few minutes to answer the survey so that I can direct us in the right direction. I have our upcoming events on the screen right now. So, this coming Saturday, June 28th, if you are in the Orlando area, it's not too late to join us for this in-person event. You can just visit our website to register and connect with other myeloma patients in your community. I'll share more about this program on the next slide, including where Connect to Cures is headed next. On Tuesday, July 1st, we'll be hearing from Diana Valentine, who is our financial advisor. She is going to share her expertise on navigating the complexities of Medicare and Medicaid to help you make informed cost-saving decisions. And then on Wednesday, July 2nd, we've got a event that is for all blood cancers. So if you have family or friends who are in different blood cancer spaces, you won't want to miss this wonderful event. It is going to be featuring Ruth, who is a myelofibrosis patient, advocate, and author. Ruth will discuss what it's like to live with a chronic cancer and how she continues to advocate for others through her journey. The month of June and July are packed with webinars and other events, so please check our registration page through healthtree.org. I'll also send that out in the follow-up email. So this is our Connect to Cures event that I've talked about a few times already today. These are live in-person events that are meant to bring resources, support, and valuable connections directly into communities. If you're located in any of the cities shown on the slide, we'd love for you to join us and help spread the word to those in your area. We've already hosted our first two events and just about to host our third, and they've been a wonderful opportunity to meet new people, connect with helpful resources, and build community. The QR code on the screen will take you directly to more information. I also dropped that in the chat, and we will put it in the follow-up email. I would like to again thank our sponsors, Adaptive, Pfizer, Johnson & Johnson, Amgen, and GSK. And lastly, I want to thank you again for joining us today and for helping to make our multiple myeloma community strong. I've got my email address on the slide here, so please don't hesitate to reach out. I love to hear from you and hope to see you again